Today, we will be discussing deconvolution, a spatially resolved transcriptomics methodology used to identify cell types in a heterogeneous mixture. First, we will discuss why we are interested in knowing a spot's content. Then we will talk about what is deconvolution. Next, we'll go into the details about the SCDC tool. After this, we will describe how the tool Seurat version 4 and SCDC identify cell types using deconvolution works in Chipster. And finally, we will list the input files, select the correct tool parameters in Chipster, and list the output files. So first, let's start with the introduction describing our objectives when using deconvolution on Visium data. To provide some background, Visium data is comprised of spots containing several cells. And our goal is to identify the cell types in a given spot, as one spot can be considered a mini bulk RNA-seq dataset, or 1 to 10 cells. One approach to detect cell types is by using deconvolution, a method originally developed for bulk RNA-seq data. So what is deconvolution exactly? This method predicts the proportions of different cell types in a spot based on the gene expression profile seen here. While there are several tools available to deconvolve a mixture, one common method is to use the SCDC tool. SCDC is a popular tool used to deconvolve or identify the components of a spot's mixture, and in this case, the cell types. Spot deconvolution is accomplished by using a single-cell reference or single-cell RNA-seq dataset to estimate the abundance or proportion of different cell types in each Visium spot. For more information on SCDC, please refer to the following websites. So how does the deconvolution tool work in Chipster? First, Chipster uses the Seurat R package to identify top cluster marker genes in the reference single-cell RNA-seq dataset. Next, we filter for genes also present in the spatially resolved transcriptomics or SRT data. Second, Chipster uses the SCDC R package, where the genes from the previous step are used to perform the deconvolution itself. After running the deconvolution step, we obtain the predicted proportions of cell types. These results are then added back to the Seurat object and can be found under the SCDC assay. Third, using the Seurat R package, the spatial feature plot is generated by using these predicted proportions of cell types. Finally, now that the Chipster tool has the cell type predictions for each spot, the tool then proceeds to do an additional step to identify which cells are spatially variable. And for this, the Chipster tool uses the mark variogram. The mark variogram method happens to be the same method used earlier to detect spatially variable genes. This data is then used to plot the spatial plot and the violin plot. So what files are input into Chipster? There are two input files that are needed. The first is the single-cell RNA-seq reference dataset. The second object to be used is the Seurat object containing Visium data, which should be subsetted for specific clusters of interest to avoid false positives. It is typically a good idea to not use the entire tissue slice, but rather the area that you expect to be matching the reference dataset. Now we will show how to use the tools Seurat version 4 and SCDC identify cell types using deconvolution in Chipster. First, we will select the correct input files and the tool itself. This is the RDS file, which is the reference dataset, and this is the R object, which is the subsetted Seurat object containing Visium data. From there, we will select the correct NGS tool category, or spatially resolved transcriptomics, as seen here. After this, we will select the correct tool, which is Seurat version 4 SCDC, identify cell types using deconvolution. After this, we will click on the parameters button, seen here, and this will open the parameters window. From here, the user can decide how many top markers are used, as seen here. The default in Chipster is 20, but ideally you should work with a larger data set if possible, perhaps 100 genes per cluster, but this may slow down the runtime. First, you will want to ensure that you select the correct input objects, seen here. After this, 4 output files are generated. So what are these 4 output files generated from Chipster? One is a Seurat R object with SCDC added as an assay, and the second are 3 plots, or PDFs, which include the spatial feature plot, spatial plot, and violin plot. This is the spatial feature plot, seen here, which plots the predicted proportions of the selected cell types. These are the spatial plots seen here, which plots the most spatially variable cell types as determined by the Mark variogram method. And these are the violin plots seen here, which plots the most spatially variable cell types as determined by the Mark variogram method. The x-axis represents the cluster identity, and the y-axis shows the cell type prediction scores. If you need additional help, please email our team at chipster@csc.fi. We thank you for listening.